guys, welcome to Bio 112 Online, your home for biology education. Today's topic, the immune system. When we talked in the last video, we were discussing the acquired immunity. And the acquired immunity is going to be the system of defenses that are slower to develop, but are able to target specific pathogens. We left off with our discussion of antigen presentation. This is where you have a pathogen that has been engulfed by a phagocytic cell. You will then have enzymes that will break down and cleave that pathogen into fragments. Those antigens will then be displayed on the MHC2, which will then be able to be presented to helper T cells. You then have that macrophage move through the lymphatic system until it finds the correct helper T cell. As it finds that correct helper T cell, it will move in close. It will then match up its MHC2 with antigen with the helper T cell's T cell receptor. If there is a match, then you will have the macrophage release some cytokines, the helper T cell will release some cytokines, and that will convert this helper T cell now into an active helper T cell. Those helper T cells, once they've been activated by an antigen-presenting cell, will rapidly divide into memory and activated helper T cells. They will then secrete cytokines that will activate B cells and cytotoxic T cells, and they will initiate both the humoral and cell-mediated responses. And that is the topic of today's video. We're going to start off with the humoral immune response. The humoral immune response, as you recall, is the secretion of antibodies and complement proteins and antimicrobial peptides in response to a pathogen. This is being conducted primarily by the B cells. These B cells are going to be lymphatic cells that produce and secrete antibodies. You recall from the previous video that those antibodies are actually the receptors for those B cells. The humoral immune response helps defend against pathogens within the extracellular fluid. In this, the B cell, as it goes about its normal routine, will come in contact with those antigens, those spit out bits of the broken up viruses or bacteria that have been consumed by the phagocytic cells. Those B cells will then go through and seek out a helper T cell. Once they meet up with that helper T cell, they will become activated and produce both memory and plasma cells. Those plasma cells are going to be the cells that are able to produce the antibodies. The antibodies are going to be a soluble form of that cell receptor. That cell receptor, once it becomes activated by encountering the epitope on the antigen, will begin to stimulate a signal transduction within that B cell. It'll also take that antigen and place it on its own MHC2 receptor. The activated B cell will now go through, as we said, and find a helper T cell. Once it does find that helper T cell, it will go through and present its MHC2 receptor to that helper T cell. If again there is a match with the proper helper T cell, then the helper T cell will begin to release cytokines, further activating the B cell. And those cytokines will stimulate the B cell to then go through the process of cell division. That process of cell division will result in copies that become memory cells, and you'll also have active plasma cells. Those active plasma cells will then begin to release their receptors into the surrounding fluid. Those released B cell receptors are the antibodies and are going to be specific for the pathogen in question. These antibodies have several major roles. The first of these is neutralization. Neutralization is a binding to a surface of the pathogen which prevents its ability to further the infection. The second main thing that these antibodies will do is opsonization. Opsonization will go through and act as a tag that will help other macrophages in recognizing the pathogens. Finally, there's a complement system. The complement system will go through and bind the complement proteins onto the attached antibodies. In doing so, they form a complex that will allow the complement proteins to bind and destroy those pathogens. For neutralization, if you have a virus infection, then the antibodies can coat those viruses, preventing them from accessing other cells. In the case of opsonization, you can have those antibodies coating bacteria. Then when you have a phagocytic cell, such as a macrophage, and encounter those, it can more efficiently identify those pathogens. In addition, you have the complement system. In the complement system, you have the antibodies that have bound on, and these can serve as anchor points for the complement proteins. Those complement proteins can then detach from the complex and can begin to form pores within the bacteria. As the bacteria forms more and more pores, it will lose its ability to maintain an internal environment that's different from its outside environment, resulting in cell death. So that's the role of the antibodies in helping to fight infection. The other main way in which your body will fight infections is through the cell-mediated immune response. The cell-mediated immune response will be the activation of cytotoxic T cells. Those cytotoxic T cells, as you recall, have that CD8 receptor, and their main job is to destroy body cells that are malfunctioning or harboring bacteria and viruses. The cell-mediated immune response involves the helper T cells again activating the cytotoxic T cells. In this case, those cytotoxic T cells will again multiply into memory and active forms, but then the active forms will go along seeking out cells that are displaying evidence of that infection. So after a cytotoxic T cell has been activated from a helper T cell, it will divide. It will then go through and start checking all of the cells in the body for the infection. Infected cells will be displaying signs of their infection on their MHC1 complexes. The T cell links it up with the CD8 and its own T cell receptor. If there's a match, then that T cell will begin to release a variety of toxic compounds to destroy that cell. The advantage here is it prevents that cell from further replication of that virus. The proteins that are used to destroy this cell are going to be in two major classes. The first of these is perforin. Perforin will go through and form pores on the target cell's membrane, causing it to leak out its contents. As it does so, then antibodies can enter that destroyed cell, eliminating any residual viruses that are still found therein. In addition, you have granzymes. Granzymes are going to be enzymes, break down proteins, and are also key in initiating apoptosis, which is programmed cell death, causing an infected cell to basically take itself apart rather than produce more viruses. The proliferation of helper T cells, cytotoxic T cells, and B cells allows the acquired immune system to effectively deal with the current infection. And it also allows the body to recognize the same pathogen in the future. But that later recognition is going to be the topic of the next video. So until then, stay safe, stay healthy, and I'll see you later. It's not my wife.